All right, welcome everyone. So today's lecture will be on genome engineering. So basically, how do we edit genomes and how do we do that really fast and in very high parallel ways. So this is the last lecture of the last module before your presentations. So this is kind of exciting. And the tools that we're going to talk about today is number one, how do we edit genomic segments in very high throughput? Basically, how do we synthesize many different versions of elements in order to test the functionality of different mutations in the genome? Number two, how do we do this in really high throughput? And number three, how do we actually edit the genome endogenously? And there we're going to talk about various technologies, including TALENs and zinc fingers, and also CRISPR-Cas9. So we focused a lot of the class on reading DNA. How do we basically understand DNA? We looked at technologies such as RNA sequencing, chip sequencing, DNA methylation profiling, high-C, and folding of chromatin in main dimensions. Something that we have not focused on is how do we write DNA? So again, being an engineering school here and being an engineering discipline, being in an engineering department, we have to think about sort of how do we create new constructs. And that involves a lot of complex tools, such as, how do we barcode, how do we create primer pools, hybridization pools, RNAi libraries, how do we create synthetic genes, mutant libraries, and so on and so forth. And the first application that we're going to talk about today is how to basically systematically create new regulatory elements. So through comparative genomics, as we mentioned in some of the earlier lectures in the class, we can identify in high resolution where are important regulatory regions in the genome. Through epigenomics, we can recognize where and when these elements are active across different cell types and tissues and conditions. Through genetics, we can map mutations that can disrupt these elements and ultimately lead to disease. But what we end up with is hundreds of megabases of cis-regulatory elements without the ability to perturb them in very high resolution and very high throughput. So here's how things have been done traditionally. So traditionally, you would basically take a putative regulatory region, sometimes on the order of thousands of nucleotides, and you would basically clone it in front of some common promoter and a reporter gene that basically tells you, is that region driving gene expression? Everybody clear as to what we're doing here? So basically what we're doing is simply testing, if I take this element from its endogenous locus and I put it somewhere very precise in the genome where I have, a reporter gene, or if I put it in a non-chromosomal element, such as a plasmid, can this region of DNA drive the expression of that reporter? Yeah. The yellow is some kind of promoter, and then here you're basically putting upstream of that common minimal promoter some kind of enhancer element that will basically dictate whether the promoter will be activated or not. Any other questions? So that's sort of the typical approach. And basically, then in order to map this element in high resolution, what you do is mutagenesis. You basically go through different nucleotides and you perturb them. And you say, well, if I delete this nucleotide, what happens? If I delete that nucleotide, what happens? If I delete that nucleotide, what happens? Sounds good? So everybody can see how this can be effective, right? If I do this systematically for every one of thousands of nucleotides, I will eventually figure out the code for, what are the important nucleotides in this particular region. The problem, of course, is that number one, this might still be incomplete because we might only get at sort of single nucleotide modifications as opposed to pairs of nucleotides that might be compensating for each other's loss. But number two, this is extremely laborious. Basically, you need to create a new construct for every one of those assays. So the bottleneck is basically how do we generate and clone individual variants, and how do we, use these luciferase assays or green fluorescent protein to basically, report the activity of these elements in high throughput. So one technology that has been particularly successful in overcoming these limitations is known by many different names, including massively parallel reporter assays, or MPRA, or MPRA. So then the idea here is that we're going to use DNA synthesis through microarrays, the same microarrays that were initially used to probe gene expression, by basically hybridizing two RNA and then seeing which spots hybridize and light up. Everybody remembers that technology? So basically, this is how microarrays are made. So the idea here is, instead of basically building a microarray to probe, only to sort of hybridize, only to use it as a sponge to capture things, what you're going to do instead is synthesize this microarray and then cleave all of the synthesized DNA segments from your microarray, and then use those as a very high throughput way to generate a very large library of fragments. Everybody with me on this? So what we're basically doing is, in the same way that microarray technology basically allows you to synthesize basically, just very, very large number of spots by adding one nucleotide at a time. And then what you can end up with is, 20 nucleotides or 40 nucleotides or 100 nucleotides or 150 nucleotides of uniquely synthesized sequence, addressable in a sort of, well by well way. Instead of now simply generating all these wells, what we're going to do is generate all these wells and then use the synthesized sequences as a pool. Does that answer your question? Are you with me? So this is the traditional way that microarray technology works. You basically create these probes that are complementary. What we're going to do now is use it as a way to synthesize a lot of fragments. So you've now synthesized all these fragments. You've synthesized a library of 50,000 distinct fragments. There's many, many copies of each of them, and every one of them has, some small error rate. Then, instead of just synthesizing the fragments by themselves, what you see, so if you want to synthesize 50,000 versions of this with high throughput mutations on every one of these elements, what you can basically do is create all of them in this red portion, and then create a unique barcode for each of them in this green portion. The fragment itself might be 145 base pairs, and then the barcode might be 20 base pairs. So you now have a unique barcode that can basically tell you about the activity of each fragment. And then within your synthesis library, you also include a small adapter that will then allow you to carry out the molecular biology to open up that region and insert a common promoter and then a common reporter gene for each of those. Okay? So I end up with, a library of 50,000 fragments, each with a unique barcode, which is downstream of my reporter gene. Who's with me on this one? Raise your hands if you're with me. Awesome. So basically, what that allows me to do now is, in very high throughput, test different versions of regulatory elements and effectively be able to engineer new elements, be able to, in very, very high throughput, test individual mutations to those elements, and so on and so forth. Okay, any questions so far? Who's not with me? Okay, good. So once you have generated that library of many, many fragments, you basically insert them, you transfect them into, a bunch of cells, and then you culture those cells, selecting for those that have, for example, the fragments based on some, selectable marker, like resistance to some antibiotic. And then you basically carry out any kind of experiment you would like. You can put, these cells into different conditions, and then amplify the resulting DNA that gets produced. Every single one of them generates a sequencing, an RNA molecule with a different version of that enhancer. And basically, enhancers that work more highly will transcribe more molecules and will have more corresponding barcodes. So what then I can do at the end is simply use PCR amplification and then sequence the barcodes. and count how many times did each element lead to transcription of one of the barcodes. Okay? So I can effectively, in one experiment, evaluate 50,000 versions of a regulatory element. Everybody with me so far? Great. So then, the question is, does this work? And then here's the evidence that this works. You can basically see that if you compare the level of expression that you infer from the counts of barcodes through this MPRA experiment, you basically see that it actually correlates very, very nicely with the amount of luciferase that you can measure using very low throughput assays for the same assay. But the throughput is now increased about 1,000-fold. Okay, so instead of doing 5 experiments, you do 5,000 experiments with the same time. So then you can apply this in many different ways. You can apply this to deeply profile a single region, or to shallowly profile thousands of regions. So let's look at both. Here's the first application of this technology by Tarja Mikkelsen at the Broad Institute in collaboration with our group. He basically looked at a synthetic promoter, which is this cyclic AMP responsive enhancer, or a CREB. And then he basically looked at, can I infer for the synthetic promoter all of the nucleotides that are important? And then he also applied it to this second element, which is this virus-inducible enhancer of human interferon beta, which is much more dirty in its design, because this was not human engineered, and it has all these overlapping binding sites. So then you can do multiple strategies for figuring out what works. You can basically change one nucleotide at a time for every base, or you could just randomly sample multiple combinations by basically synthesizing pairs of random mutations. So, the first thing was to actually show that this is actually reproducible with different mutagenesis strategies. And then asking for every single nucleotide in this sequence, if I change it to an A, if I change it to a C, if I change it to a G, or if I change it to a C, how does the corresponding activity measured by the barcodes change? And what you can see here is that if the first is a G and I change it to a G, well, I'm not going to even measure that. If the first is a T, or if the, if the second one is a C and I change it to a C, again, no change, because, one of the 4 will always be preserved. But then you can ask, if I aggregate across all of them, basically, which bases change the most? And you can see here in red, the nucleotides that show the highest change in activity are in fact the ones that perturb the known CREB binding sites in this element. Right? You can see here this dramatic increase if I perturb the 4th one, and if I perturb the 3rd one or the 2nd one, regardless of how I perturb it. Is everybody with me as to what we're measuring here? And then you can aggregate across all of the different nucleotide versions, and then sum up the data, and then ask, well, what is the relative importance of every one of these elements? Okay? And then that pretty much recapitulates the native activity. And then the same thing for the second promoter, the interferon beta promoter. You can basically see here that a lot of these changes that have the most impact are in fact overlapping with this known motif. And then when you sum them all up, you can in fact recover this quite accurately. Okay? Everybody with me so far? So again, this is an approach to not only write DNA, but to write it super, super, super fast and in very, very highly parallel ways, in order to then be able to dissect regions by high-throughput mutagenesis. But we'd like to go beyond that. We'd like to go to synthetic biology. We'd like to design new classes of elements. So for example, what you could do is figure out ways to optimize the elements and lead to even higher expression. And using what we learned previously, we could actually go and synthesize additional binding sites and show that indeed this greatly improves the efficiency. And then what you can see is that 52 out of the different variants, in fact, showed either similar or higher increase to what you could engineer based on your knowledge. You could also use this technique for scanning different regions. So for example, if you have a very, very large region of 500,000 nucleotides, you could basically synthesize all of them by tiling that entire region and then ask what is the activity that they obtain for each of those. So by tiling the entire region, you can then say, you know, great, these are the regions that show the highest activity in a reporter construct. And indeed, these fall quite often in DNA hypersensitive regions, in you know, regions that are marked with enhancer marks, such as H3K27 acylation and H3K4 monomethylation. And you know, they do not as much fall in promoter marks, such as H3K4 ME3. So then you can, you know, go in and further increase the resolution of these regions and then sort of get down to very high resolution mapping. And what you can see here is that this region is in fact corresponding to an evolutionarily conserved stretch of nucleotides, which is DNA's hypersensitivity, which is accessible to DNA, and which actually falls right in the middle of this H3K27 acylation peak. And not only does it fall in the middle of that peak, it falls specifically in a dip within that peak. What does that mean? Well, all of these histone modification marks are measured by pulling down nucleosomes. That's where the histones are. So the histones are basically making up the nucleosome. And when I see a modification, that basically means that a nucleosome is sitting exactly there. But what happens with actual transcription factor binding? The transcription factors bind in between the nucleosomes and they actually push the nucleosomes away. So what you can see here is that this binding site falls exactly in the middle of two well-positioned nucleosomes that are now pushed to the side, and then the marks are deposited flanking that driver nucleotide where the binding is happening. Everybody with me here? Raise your hands if you're with me. Awesome, great. So then, you know, you can do a second round by basically creating random variants of that region and then figuring out in high resolution the footprints that contain the nucleotides within this region, and then, you know, analyze them further. So that was one approach of basically going very, very deep for one region, whether it's a single, 500 base pair region, or whether it's a very, very large region, which you can then do hierarchically. An alternative approach is to basically test in a motif-guided way thousands of regions. So using exactly the same technology, we can basically predict who are the regulators that are functioning in different cell types. So looking at the enrichment of particular motifs in the sites that are active in HepG2 or in K562, two different cell lines, one immune line and one liver line, hepatic line here. You can basically see that there's different regulators, hepatic nuclear factor, as well as GATA and NRF, which are immune regulators. And then there's also repressors corresponding to each of the lines, which are basically maintaining the sites that correspond to this motif off in these corresponding cell lines. Everybody with me so far? So then what you can do is basically go driven by these nucleotides, instead of testing thousands of variants of every one of these regions and basically perturbing every nucleotide, you could perturb specifically the nucleotides that correspond to your computationally predicted binding sites of driver motifs. Everybody with me so far? Great. So you can basically, you know, test a wild type sequence with 20 barcodes and then test a scrambling of that motif or a removal of that motif or a single base pair change of that motif that either maximally decreases the PWM, the position weight matrix of that motif, or makes the smallest change, or maximally increases with a single base pair, or just random base pairs to basically see what happened. And again, you include these sequences in front of a common promoter, a common reporter gene, and a different barcode for each element, 20 different barcodes for each element, and then you transfect both one cell line and the other cell line. So here's what the results look like. Here's one example of a region which, based on its chromatin states, is active in HepG2 and inactive in K562. So, there's a locus here which is being tested. You can see here in high resolution that this region actually falls in a dip in this chromatin profile, again corresponding to the internucleosomal region, whereas it's sort of purely flat in the other cell type. And now, if you perturb that motif, what you see is that, first of all, for wild type, the expression is much higher of the reporter gene in HepG2. Why? Because although the transacting factors, the regulators that are active in HepG2, are in fact on, whereas the regulators that are active in K562 are not driving expression exactly as you would expect based on this endogenous epigenomic information. So we're now using the endogenous epigenomic information in the context of the nucleus of that chromosome to basically predict what would happen based on the endogenous marks. And what we can see is that when we remove this element from its native endogenous context, it is still able to drive expression in a plasmid outside the chromosomes to, drive expression of the reporter gene in HepG2, but not in K5. Everybody with me here? So then, if we scramble, remove, or maximally decrease the motif, that goes away. If you do non-disruptive changes, such as the least change or the maximal increase, you again see, continued activity in one cell type, but again, continued non-activity in the other cell type. And then if you do random changes, it kind of depends as to whether you disrupt the motif or not. And in this particular case, the motif is an HNF4 factor, hepatic nuclear factor 4, which is known to be active in these hepatic cell lines. Kind of cool, right? Like, biology makes sense. So then you can do this across many different sites, and then you can see that the wild type, performs the best. The maximal increase is the highest. Then, the random changes depend on whether they perturb or not, and so on and so forth. Okay, and then this works across HNF1, HNF4, FOXA, GATA, which are all predicted activators in the matched cell type, and then NRF2 as well. And then for repressors, what we basically see is that when you perturb a repressor, sometimes you see actually increase in activity, which suggests that perhaps the repressor is what was keeping things inactive, but now by knocking out the repressor motif, you basically have de-repression of that element. So that's all nice and good, but basically this technology, which allows us to test 50,000 variants at a time, can only be used to either test one site really, really deeply, or many, many sites really, really shallowly. What we'd like to do is both. So what we're going to do next is look at next generation applications of this technology to basically, overcome these limitations. And then the key idea here will be the following. Instead of testing 20 barcodes all at exactly the same position, what we're going to do is test one barcode at each of many, many different positions. So we're going to basically make a design which tiles a particular region at, say, 5 base pair increments. Okay? So that allows us to now test a larger segment because our array-based synthesis only allows us to test 145 base pairs at a time. We could previously test only very small elements, whereas now we can actually test larger elements and then, scan across a particular region. The big advantage here is that we will be then able to compare the activity between neighboring sites to basically say, aha, these 5 base pairs seem to now increase the activity when included, thereby enabling me to pinpoint in high resolution where are the driver nucleotides. And I'm going to, focus a lot more on that. So basically, this is akin to perturbations, whereby I include or exclude individual 5 base pair elements, but without actually going through the experiment of perturbing these elements. So to apply this technology, we selected many different regions in the genome. We selected them proportionally to how often these were DNAs hypersensitive. And that led to an enrichment for enhancer regions and, CF binding sites and so on and so forth. But we specifically excluded, promoter regions. So we used 2 different designs. One was the high resolution design that I showed you earlier. The other one was a low resolution design where we use, again, 10 different barcodes per sequence, but now we only use 30 base pair offsets just to test the technology. And what we found is that indeed, as you sort of got closer to where we thought the signal was coming from, the activity became stronger in the matched cell type. but not in the non-matched cell type. And, you know, this was reproducible basically between replicates. This was a very strong agreement. But by offsetting, even by one, 30 base pair chunk, we saw that there were, in fact, very strong effects. So this is testing the exact same tile. This is testing consecutive tiles, 30 base pairs apart. And you can see that indeed we can have a dramatic difference, probably driven by these regions here in white that are distinct between these elements. Who's with me so far? Raise your hands. Awesome. So then what we can do is basically look at specific examples as we're tiling different regions. Boom, activity goes high here. And indeed, in that 30 base pair interval, we could find post de facto, after the fact, a motif for the corresponding regulator, which basically was quite reassuring that indeed the biology made a lot of sense. So then we use that to discover unique motifs within these regions and so forth. But then the most interesting part was this very high resolution design, because it allowed us to now tile much larger regions, to tile, at much smaller offsets, to center on the peaks, and then to use a single barcode per peak, thereby optimizing the barcodes, creating more elements per array. And we used a larger array as well, and we tested all 25 chromatin states. So that basically allowed us to now go a little higher throughput here. And to do that, we basically needed to develop a computational model that allowed us to effectively deconvolve the activity of all of these overlapping segments, because every single experiment is measuring 145 base pairs into 5 base pair increment activities. And then this is just a big system of linear equations whereby the sum of all of these is in fact corresponding to each of the measurements, which then aggregates, 29 sites for most of them, and then, fewer and fewer sites in the middle. Okay? Everybody with me here? So then, we made some assumptions about the underlying distributions of, the observations, the measurements, the mean, the variance, and so on and so forth. And then we tested elements by, again, overrepresenting enhancers, weak enhancers, CF binding sites, and so on and so forth. We basically tested things using a minimal promoter, a strong promoter, using 2 different replicates, each array, capturing elements from many cell types, and then each tested into different cell lines. And what we found was quite remarkable, that as you scanned across this sequence, you could basically, in high resolution, infer where the binding sites were coming from. You could basically see dark, 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 dark coming into, high activity, and then high activity coming into low activity. And that allowed us to predict both activating elements, where the high activity was, appearing, as well as repressing elements, where the low activity was appearing. And those, in fact, corresponded to known regulatory motifs when motifs were previously known. So, here's one example using at 60 different sites that contain that particular motif. You can see here reproducibly that as we're tiling, we're finding the strongest activity exactly where the motif is. And here's an example of a repressive site, NRSF, this neuronal repressive factor, which basically represses every cell type except neurons with this very long motif. And then you can see that whenever that motif was found, activity was shorter right in the middle. Who thinks this is kind of cool? Awesome, good. So basically you're now able to sort of, without changing the technology per se, simply by applying the technology differently, test both many, many sites and high resolution. Basically, we're now able to test 15,000 different regions in the genome, tile each of them at 5 base pair resolution. So basically, we've sort of accomplished what we wanted of sort of combining both broad application and deep application in each of the regions. And then we could do all kinds of things in terms of discover motifs that were common between the cell types. For example, we found that this motif led to the highest activity. And in fact, nobody knows what this motif does. This was discovered independently in a bunch of different experiments as the most enriched site, but we still don't know what the regulator that binds it is. And again, using this in a completely agnostic manner allowed us to discover that. We're able to find sort of repressors, activators, able to find cell type specific motifs. So these are off diagonal elements, which are high in one cell type but low in the other, or high in one cell type and low in the other. And then these are again involved in cell type specific regulation. We're able to show that activator motifs are the most enriched when the regulatory score is the highest. Repressor motifs are the most enriched when the regulatory score is the lowest. That the most active and the most repressive segments were in fact evolutionarily conserved. And we were also quite surprised, but in retrospect it makes a lot of sense, that some of the most active sites were in fact driven by repeat elements. So repeat elements, as you know, have invaded the genomes of many different species, catalyzed their own transcription, enhancing their own transcription by basically recruiting regulators of the target host cell. So basically they show up, they use motifs that are very, very strong for that host cell, basically recruit the machinery of that cell and then catalyze their own transcription. In this particular case, you can see why these were so optimized for enhancing their own activity, that in fact they learned how to create motifs that are the most active in the host cells. Everybody with me here? So some of the most active elements were in fact the most enriched for repetitive regions. And these differ greatly from chromatin state to chromatin state. And we were able to discover all kinds of cool things, such as repressive motifs in the context of otherwise active regions, and so on and so forth. So this was great. We were able to go, instead of testing only 2,000 sites in a fairly shallow way, we're able to now test 15,000 sites much, much more deeply. Instead of doing one deeply, we're now doing 10,000 deeply. And then what we basically said is, hey, can we increase that by another few orders of magnitude, by another 3 orders of magnitude? Can we, so MPRA was already 3 orders of magnitude more effective than traditional luciferase reporter assays. This was already 3 orders of magnitude more effective, or 4 orders of magnitude more effective than MPRA, by basically being able to test 10,000 sites in high resolution rather than only one site. And then what we wanted to do is go another 3 orders of magnitude beyond that. So how is that possible? Well, the first thing we need to do is forego synthesis, because synthesis is a rate limiting step. We're basically taking all these DNase hypersensitive sites, selecting them, synthesizing them, and then testing them. So we basically said, hey, wait a minute, what if I get rid of the synthesis altogether? So what can I do here? Well, instead of first doing a DNA accessibility experiment, such as DNase hypersensitivity or this assay for transposase accessible chromatin or ATAC-seq, what we basically said is, let's do ATAC-seq, but don't do just seq after ATAC. Simply cut out the accessible regions from the DNA and then create a library from the accessible regions directly. So instead of sequencing the accessible regions, synthesizing them in our computer, ordering an array, and then getting back the array and then plating it, what we basically said is, let's skip all that step. Let's simply synthesize, let's simply capture accessible region rather than resynthesizing. Who's with me on that step? It's a key step. Great. So then once we've captured this from the genome, we can basically, insert it directly into reporter constructs using a technique introduced by Alex Stark in his lab called STARR-seq. What does STARR-seq allow you to do? Instead of the traditional approach where the barcode is synthesized separately and inserted downstream of the transcribed element, what STARR-seq says is, wait a minute, we know that enhancers can enhance transcription both upstream and downstream of the transcription start site. So why don't we put the enhancer downstream of the transcription start site, and in fact, even downstream of the reporter gene? So what we're going to do is insert the elements in the untranslated region, in the 3' untranslated region of the reporter gene. And then they will catalyze their own transcription. Who's with me on this one? So this is the concept of STARR-seq. It basically says, let's use self-transcribing elements that basically that catalyze their own transcription. And then that means that I can use PCR here flanking the insertion site to use the barcode, the elements themselves as the barcodes. Sounds good? So what we're ready to do now is increase this by several orders of magnitude again. So because we have no synthesis, we can capture 7 million fragments at a time. And this is only a matter of how deeply we want to sequence. There's no synthesis. Because we can simply size select, we can test much longer fragments. Because we're selecting accessible DNA regions, we have very high sensitivity. Because we integrate in the 3' UTR, they're self-transcribing, so we don't need a barcode. And because these elements are now going to be very densely overlapping in these accessible regions, we can actually tile the regions in the same exact way that we did before, and then infer from the tiling pattern where are in high resolution the driver nucleotides. So putting it all together, we can basically test millions of fragments in a single experiment with high sensitivity and high specificity, and we can infer in high resolution the driver nucleotides in each of these regions. This is what the data looks like. So after you've basically mapped these elements back to the genome, you can sort of see their activity, higher activity in yellow here, and you can see how they're tiling the region with random starts and random ends. But you can see that the higher activity is in fact coming from the center region, which indeed contains evolutionarily conserved elements, and indeed contains binding sites, including for RONX3. Who thinks this is kind of cool? All right, so basically, this is just using principles that we've learned throughout the class to sort of engineer new technologies that allow us to test sites at dramatically high resolution. If you look now at the DNA library, we're basically capturing these accessible regions, and we're putting them into plasmids. If we sequence the DNA plasmid library, what we see is that the most elements are in fact coming exactly from where hypersensitive sites are, whether you measure them by ATAC or by DNAs, right? So basically there's this huge representation of these accessible regions. You can see that the length of these elements is in fact much longer than what you would be able to test otherwise. So the median region length is 1,300 nucleotides because we're tiling it with many elements. And the median element length is in fact 337 nucleotides, much larger than what you're able to synthesize with MPRA, which only synthesizes elements on the order of 130 to 200 nucleotides. So that means that individual elements are larger and the regions tiled are much larger. Moreover, because we're capturing regions that are accessible, we're able to enrich for multiply coupled regions by several hundredfold. For active enhancers, for active promoters, for active peaks, and so on and so forth. This is highly reproducible, 0.95 correlation for these high activity fragments, 0.75 correlation for low activity fragments. And then if you look at a region that was previously tested laboriously using luciferase assays, they basically tested all these elements shown here at the top, and then the red ones among them were the ones that showed activity. You can see here that our DNA input is in fact capturing all these elements and therefore testing them. And then our output is perfectly recapitulating this luciferase activity assay with 88% Pearson correlation. Okay, so that basically means that even though we've tested 7 million fragments across the genome, for those 20 that were in that region, we actually have very high reproducibility with luciferase. We find that motifs are enriched, these motifs can be clustered, that we can now start inferring these elements in high resolution. We had to, in fact, write a new algorithm for inferring the high resolution activity because Sharpr, the original algorithm, was assuming 5 base pair increments. We then developed Sharpr RE for random endpoints, which basically allows you to do that even when the endpoints are randomly inferred. And then that allows us to now capture very high resolution elements across 7 million tested fragments. That allows us to also, because we're actually sequencing the DNA, capture polymorphisms. So basically, whether you have an A or a G at that particular location will actually lead to differential activity in this reporter assay. And we can, in fact, capture that directly. And indeed, this is consistent with QTLs from allele-specific activity. So this paper was, in fact, just accepted. It will appear hopefully within the next few weeks. We sent the proofs yesterday. So this is, again, one of those cases where, this class is moving very fast. I couldn't have given this lecture, last year because this work was not even done yet. So, in trying to keep up with the latest progress in science, here's a preprint, something that's not even published yet. All right, so this is all synthesizing elements and then testing elements in very, very high throughput, but outside their endogenous locus. This is also, this can be modified to basically, instead of transfecting these elements with plasmids, transfecting cells with an integrating vector that will then go into an endogenous locus. But the next stage in all of that is to not just test elements that I synthesize, but to actually therapeutically perhaps go in and change nucleotides in the genome. So what I'm going to talk about next is these technologies for cutting and editing DNA that have been, very, very popular recently, including, TALENs and ZNFs in the last, decade, and then CRISPR-Cas9 in the last, few years. So what is the whole point of genome editing? How, what is the whole principle? The concept there is very simple. It's what George Church calls genome vandalism. So you're not editing DNA really. What are you doing? You're cutting DNA. The whole point of genome editing is that we're going to first just brutalize DNA and just cut it. Basically, take the location where we want to do a very minute, fine, fine resolution edit, and we're just going to first cut that region. That sounds a little counterintuitive. Why are you vandalizing it? And the answer is that there's a repair machinery in the genome, in every cell, that basically will look for these double-stranded breaks and try to repair them. Okay? How does it normally repair them? It normally repairs them by basically looking elsewhere in the genome for the homologous chromosome. It basically says, oops, I lost dad's copy, no problem. Mom has one, I'm going to look for mom's copy. Okay? And vice versa. So basically, it scans the genome, it looks for regions that are homologous to this part and to that part, and then it uses them as a template to synthesize the missing piece of DNA. Who's 100% with me here? Raise your hand. Awesome. Great. So there's many different things that can happen. The first is what I just mentioned, homology-directed repair, where you basically find some, repair template, which is homologous in the genome, and then you basically lead to a reparation of that site. If you just throw, double-stranded break to the genome without a repair template, it will try to find the homologous chromosome, or it will find some kind of paralogous site in the genome from an ancient duplication event where that region is found in multiple copies. It might just choose a template from one of the other copies. There's no guarantee that it will be from the homologous copy. But one way to trick that machinery into doing whatever edit you would like is to actually feed it a lot of template DNA for repair. You basically say, well, I want to change this endogenous site from a G to an A. Let me just first brutalize it and then throw a bunch of versions of that little piece of DNA with the alternative version. And when it's looking frantically to find something to repair it with, some of the time, it will basically capture, the one that I gave it and it will repair it. Okay? Who feels that they've learned something here? Yay, great. That's one pathway. That's homology-directed repair. That's when we get lucky. The other pathway is non-homologous end joining. So basically, the genome sometimes says, well, listen, I don't care so much as to what's there, but I care a lot about the integrity of my DNA, the integrity of my chromosome. So what I'm going to do is I'm going to just simply, chop them off a little bit to basically sort of create a nice blunt site, and then boom, glue them together. And that's basically creating an indel mutation, which sometimes will create a premature stop codon if you fall inside a gene, sometimes will create all kinds of other havoc, and sometimes will be simply benign, or sometimes will simply inactivate whatever gene you want it to inactivate. So basically, this double-stranded break can be used to either knock out a gene or knock out a regulatory region, or actually repair it to something that you would like. Okay? Everybody with me so far? Any questions on this? Yeah. Yeah. So when you flood it and homology-directed repair happens, it will typically use one of the things that you flooded it with. But this pathway is only used a fraction of the time. And it will successfully repair only a fraction of the time. So the way that this is usually done is that you can select cells by visualizing somehow which ones sort of captured the repair that you had, prompted them with. And then you can basically either capture a bunch of cells and then look which ones worked after the fact, or have some assay for selecting the cells that worked, or engineer constructs that are allowing you to self-select them. You can basically create a two-step process, for example, where you first have a selectable marker that indeed the break happened, and then you could have a selectable marker that says, indeed there was a repair, or indeed this was captured in there. Or you could basically say, well, let's only worry about the cells where there was a repair, and then do a first round of a selectable marker where, if this was, where you're basically selecting for one drug, where if you're missing that element, you will die with that drug. And then you take away that drug, and then you do a second round where you now excise and remove, the extra construct back into a single base pair. And then that construct could create both a positively selectable marker and a negatively selectable marker. So now you can include a second drug where if the cell was not successfully excised out into that single nucleotide, you could, do that. So basically, there's a lot of research right now into sort of how do we make this more efficient? How do we increase the fraction of cells that are successfully edited into this particular pathway? Yeah. Well, it varies greatly, and it, varies from lab to lab. It varies depending on how you do the reactions and so on and so forth. But, it's on the order of, let's say, 20% or something like that. You have to look through 20% success here if you're, if you're doing well. Yeah? Oh, thank you. Yeah. So, again, it's an active area of research. I would, basically, if you have ideas about this, go work on it, because that's something that the field needs greatly. Okay, great. Everybody with me on sort of genome editing? It's really genome vandalism plus repair. Okay? And then the big question is, great, well, how do we create these artificial double-stranded breaks? So what you can use is programmable DNA nucleases. These are proteins that go and cut the DNA. And there's two different families, or three different families. There's zinc fingers, there's TALENs, and then there's CRISPR. Okay? So CRISPR is not a genome editing technology. No, it's a very effective way to guide cutting in, a programmable way. Okay? So we're going to get to CRISPR shortly, but first, let me introduce how was this field created, which is, ZNFs and TALENs. Okay? There's a lot of different ways that you can target a particular nuclease to the genome. You can basically customize DNA binding proteins and then send them off to cut the genome. You can use helix turn helix domains, zinc finger domains, leucine zipper domains, ring helix domains, and so on and so forth. And all of these different domains are able to now guide this cutting to specific sites. What is the challenge here? A helix turn helix domain has only 8 nucleotides that it targets. And that basically means that I'm going to cut thousands of places in the genome because I won't have enough specificity. So what we need is guide regulators that can go into a single nucleotide, a single place in the genome, if you want to target a single nucleotide from the 3 billion places in the genome that it can normally go, okay? And that's what this field has been sort of so plagued by. So then let's go through what was first discovered to do that. So the first iteration of this was zinc finger DNA recognition domain. What are they? Zinc fingers are basically one type of DNA binding site. It's one type of regulator. It is naturally occurring. But then these zinc finger arrays is something that the genome developed to target things with extreme specificity. Why? We frankly don't really know, but it did. So we're exploiting it. And then the idea here is that every one of these zinc fingers targets 3 or 4 overlapping nucleotides. Basically, one zinc finger here will, bind GCG. Another zinc finger there will bind GGT. Another zinc finger there will bind GCG, and so on and so forth. So, here's the first finger, the second finger, the third finger. And what you end up with is these proteins that basically have, all of these zinc fingers that are arrayed back to back in order to create a sufficiently large site that it is unique in the genome. Who's with me on ZNFs? Raise your hands. Awesome. Great. So basically, the challenge with ZNFs is that these motifs are very highly overlapping. Basically, you have 3 nucleotides at a time, and you have to design them and engineer them in the way that you can, target your target site of interest. So there's been a lot of work in optimizing them and making them work better by basically using these, 2-stage, these sort of bidirectional cleavage approaches, where you have one zinc finger array here, another zinc finger array. Both of them are sort of working together to cut the part in the middle. There's been a lot of work in generalizing the selection of these binding sites, basically given these overlapping selectivities for these zinc fingers, how to basically create general arrays that can allow you to bind, pretty much any motif in the genome. And these have actually been successfully used to, carry out genome editing. So they were used in knockout rats, they were used in embryonic stem cells, they were used in, human ESLs and human iPSCs, basically create edits every single time, sort of guiding, all of this molecular machinery using these zinc finger domains. And also in human hematopoietic stem cells, enabling you to now create a reproducible, supply of blood cells that have, an altered site. And this was all over the news. I mean, if you look at the New York Times in 2009, in a new way to edit DNA, hope for treating disease. I think you're reading an article about CRISPR-Cas9, then you look closely, it says, the technique, which depends on natural agents called zinc fingers. Oh, okay, well, I guess, CRISPR-Cas9 was not the first time that people thought we were gonna, edit DNA and change the human race and all of that. Here's another article here. Different companies started by basically saying, great, we're going to use the zinc finger nuclease technology. Again, the date here is 2010. So CRISPR hadn't been there yet. So that was the first technology, zinc fingers. The second technology is TALENs, basically these transcription activator-like effector nucleases, or TALENs. What they are is dimeric transcription factors with a nuclease, which are built from these individual modules that are about 33 to 35 amino acids long. Every one of these modules targets a single nucleotide. So while zinc fingers were targeting 3 nucleotides at a time, and then you had all these challenges sort of, engineering them to target any general site, what TALENs do is in fact target a single nucleotide with each module. So then the key engineering idea is that by assembling arrays of those, we can then target nearly any sequence. Everybody clear on the difference between zinc fingers and TALENs? Who feels that they're learning stuff? Yay, great, awesome. Good, so that's what they are. They basically say, great, I'm gonna build all of these arrays to basically gain specificity, and then these are gonna be guiding me to very specific sites in the genome, and then I'm gonna do the same exact thing, genome vandalism followed by repair. So there's been, a series of improvements on the technology, basically first breaking the code of DNA binding specificity for tau type 3 effectors, and then, sort of figuring out how I can engineer any binding site that I would like in the genome, and then how I can find naturally occurring tails that basically, target much longer nucleotides, nucleotide stretches, which I can then use to sort of select targets in the genome. And then design them de novo, where I can actually just engineer in the lab you know, these 33 amino acid domains that can sort of, you know, target any stretch of nucleotides of interest. Okay? And then the crystal structure was solved, and you can see these massive, massive protein looping around the DNA, each of which is now recognizing a single base. So this was actually very effectively used in plants. This was then effectively used in human cells. And then, you know, this was used to start, you know, targeting the genome and editing the genome and then cutting the genome and engineering the genome, you know, increasing the efficiency, establishing homologous recombination, you know, overcoming the assembly challenges for these, you know, very long TALENs using synthetic biology and so on and so forth. And then, you know, improving their efficiency, their capture, and so on and so forth. Okay? So this was all used to now be able to guide all of these molecules to the genome and induce these particular pathways. But then you don't want to just, you know, carry out cutting. You want to do a diversity of things. So instead of always simply chopping up the DNA, what you can do is remove the nuclease that actually cuts the DNA from this whole machinery and simply use this machinery to bring almost anything to the genome. So what else could you bring? What you could bring is a transcription factor that, for example, will stop the progression of RNA polymerase. So you could use that to then inhibit the expression of a particular gene by basically using this approach of targeting the DNA and then bringing in, you know, these very, very strong binders that will then not let RNA polymerase go through. You could also use it to start competing with endogenous transcription factors. So if a transcription factor normally binds a particular location here, what you could do is simply compete with that transcription factor, bind that location more strongly, and just not let the transcription factor bind. So then you could sort of either stop the repression of a gene or stop the expression of a gene. Everybody with me so far? You could also induce epigenomic modifications. You could bring DNA methyltransferases to methylate a particular region, or, you know, to remove the methylation. What you could do is recruit, again, DNA cutters, as we mentioned earlier, or you could also recruit a recombination factor or an integration factor that basically always puts in something in the DNA or always induces some kind of recombination, sort of change the context of a particular element. You could also recruit regulators. You could recruit a repressor. You could recruit an activator that will then, you know, either stop the expression of a gene or turn on the expression of a gene. Or you could induce specific histone modifications. All of this is genome engineering, and we haven't even talked about CRISPR yet, right? So I want you to really recognize that this is all sort of a technology that has been coming for 2 decades now in terms of the modular design of all of these different techniques that you can use to use everything we've learned about in the class to then target DNA and then carry out different steps on that DNA. Everybody with me so far? Who's kind of disappointed we haven't even talked about CRISPR yet? No one. Yay, that's great. And that was the whole point. Now, CRISPR. So what's CRISPR? CRISPR is pretty awesome. So CRISPR is basically this very cool way of targeting anything in the DNA without designing proteins, by designing DNA directly. So we're working on nucleic acids, and that's the whole point. DNA is letters after letters. What we'd like to do is, instead of having to create these crazy protein domains that will then loop around the DNA in all these complicated ways to target the cutting any place we want, what we would like is to have a very fast, efficient, super high resolution way of targeting any place in the genome. And that's what CRISPR is. So CRISPR, again, came from decades of basic biology work. Nobody was expecting this to be this humongous revolution it has been, and very righteously so that it has been. So it was first discovered as, this, very weird mechanism of resistance of bacteria against viruses, okay? So this CRISPR was providing acquired resistance against viruses. And then, how it was, a huge challenge to basically figure out how, until again, people started realizing what are these small CRISPR RNAs doing and how are they guiding it, and how, all of this is working, until the editing eventually started happening, moving to human. So why is CRISPR called CRISPR? So anybody knows what it stands for? Yeah, cluster of repetitive, interspersed, short. Good. So basically, what this is basically saying, so this was discovered bioinformatically. And in fact, Max Wolf, who's one of the students in the lab that you guys interacted with through your mentoring session, is the son of the person who actually recognized this in the first place from a purely computational analysis. So his mom was basically looking for these, trying to understand what are these repetitive elements doing in these bacteria. And she found that there were these repeat elements that were always the same in black. These spacer elements were variable, and they were each time different. And she said, what the heck is going on? And what she realized is that when she started blasting these colored elements to the NR database, the non-repetitive database, she basically saw that they were mapping to viruses. And from that moment, she said, aha, this is a viral defense, this is an antiviral defense mechanism for bacteria. Why? So she basically postulated that the bacteria were remembering in these colored little elements who invaded them and who fought, them, and then they would save an adaptive resistance mechanism to then be able to go and fight against that virus when it attacks them. So it was an adaptive way of doing so. How? By basically cleaving off the viruses whenever they come in, and then saving, like trophies on their wall, segments of those viruses that they could then use to target these viruses when they come back. Everybody with me so far? Who knew this already? Who feels that they've just learned something? Yay, awesome, good. So this is the whole point of CRISPR. CRISPR was basically this very effective way that bacteria evolved for using DNA to target DNA and to cut it, right? They would basically save these target, sites. Whenever the virus would release its DNA inside the bacteria, they would just go in and just cut that site. And that's where, the genome engineers basically went, oh my goodness, this is the thing to use to programmably cut DNA. And of course, it was a huge, huge undertaking to be able to actually achieve that. But the whole point of all this is that it's an RNA-guided nuclease. How? By basically using these, RNAs that are produced by these elements to then look through the genome, through the cell, for these DNA that would match these sites. And here's the key idea. The key idea is that there's a scaffold that gets built that guides this whole machinery. And the scaffold is in fact, inserting into the DNA and then scanning the DNA for a match between a single-stranded DNA that's open and a single-stranded RNA that's serving as a guide, to then lock onto that piece of DNA and then start, cutting it. The whole machinery is relying on proteins that will then guide the recognition and then cut off the DNA, okay? The targeting itself is guided by an RNA guide, but then the rest is a protein machinery, okay? So when we talk of CRISPR-Cas9, CRISPR is this whole sort of tandem repeat system for remembering. And then Cas9 is the protein that actually catalyzes this guide recognition, cutting, and the rest. Okay? So what Cas9 does is that it scans through the genome and it recognizes this NGG positioning signal. Okay? It's a little constrained that it only works when it has that. But that's Cas9. Bacteria are extremely diverse, and there's many, many different proteins that can be, now tested for whether they don't have that constraint anymore. But basically, the quick, rule of thumb is that if you want to edit something in the genome, if you want to target something in the genome, you better have a GG nearby. And you better have that target within some distance of that GG. Okay? But GG is pretty short, so it happens pretty often. And then once you've locked onto the DNA using this GG, then you open up the DNA, you invade the double strand by opening it up, and then hybridize your guide RNA to one of the two strands, and then using that to lock on and do your recognition. Who's 100% with me so far? Hey, great. All right, so then you basically first do a nick. You basically, Cas9N basically nicks the DNA. So a nick is not a double-stranded break, it's a single-stranded break. You only cut one part. And then what you can use is do two nicks to effectively, chop up the DNA with long overhangs that then allow you to do all kinds of additional molecular biology there. So then after this was discovered, there was a huge, huge effort in optimization, basically bring CRISPR into mammalian cells, basically, trigger the non-homologous end joining or the homologous base repair, to basically minimize the system, to make it more efficient, to basically, guide with, a very effective way, finding different Cas9 homologs, enabling, targeting, cleaving endogenous locations in mammalian cells, and then, effectively carrying out this targeting. So there was a huge, huge amount of work by, researchers in Europe, in Berkeley, at MIT, all sort of, working in parallel, trying to sort of push this system to editing. And then demonstrate that indeed you can have very precise sort of, cleavage of DNA, that you can have sort of single base pair resolution and specificity in the targeting, that, you can sort of recognize what are the specificities of Cas9, and then sort of finding new enzymes, optimizing existing enzymes, and then, creating these chimeric RNAs that can basically use these two different guides that the original machinery needed into a single guide RNA, and then improving the specificity using different offsets for these single nicks, to increase specificity, understand the spacing requirements for doing this nicking efficiently, improving the specificity of this, wild-type Cas9, so basically using different optimized constructs to increase the efficiency. You can see here that the wild-type is only at 3%. With the optimization, you can get upwards of 30%, and also increase the sort of the reduction in off-target activity. So basically, make sure that it only cleaves where you want it to cleave. And then sort of demonstrate that it works in mouse, demonstrated that it works in human, and then designing tools for selecting your guide RNAs in order to be both very highly efficient in the regions that you're targeting, and also, have few off-target effects. So then there was also a big rush for discovering other nucleases beyond Cas9. So for example, CPF1 is another CRISPR-associated single effector DNA nuclease that has, all kinds of additional properties, enhancing Cas9 specificity, discovering molecules that cut RNA rather than DNA. So some of these bacteria, instead of fighting against DNA viruses that release DNA into, their cells, they're fighting against RNA viruses, which basically are single-stranded RNAs sometimes, or sometimes double-stranded RNAs. So basically, there are indeed enzymes such as C2C2, which are CRISPR-associated, but unlike Cas9 that recognizes DNA, these guys recognize RNA. So instead of now editing the germline, which makes a lot of people happy, or editing even the somatic line of DNA in individual cells, what you could do is actually edit RNA. And then, what this allows you to do is now, have a whole new set of targeting approaches for RNA editing. And then you can see, this huge diversity of CRISPR-associated RNA effectors. and sort of how they work. And there's many CRISPR associated nucleases that can cleave mammalian DNA. There's Cas9 from different species. There's CPF1, and they have, you know, different PAM localization properties. You know, here the PAM is 3' of the target, here is 5' of the target. They have different PAM sequences. You know, the original Cas9 has this NGG. Others have, you know, different specificities. So if you have a TTT, but not a GG nearby, you might want to use this one instead. Whether it cleaves its own CRISPR array, what kind of cuts it creates, whether it's a blunt cut or a staggered cut, and, you know, whether it's consistent, how it fits into these sites, and, you know, whether it needs long or short guide RNA. So all of this has been genome vandalism, as I mentioned earlier, okay? But you don't have to do that. What you could do is couple to this Cas9 machinery cytidine deaminase, for example. I've mentioned this before, how deamination of a C can turn a C into a T, right? So if you do that, what you can basically do is never induce a double-stranded break, because double-stranded breaks can instead go to, you know, different pathways. They could lead to, you know, this non-homologous end joining, or they could repair with the wrong template, or they could just lead to breakdown of the DNA altogether. But instead, by coupling Cas9 with something that will not cut the DNA, but instead edit a single nucleotide, what you can basically get at is true base editing. So this is one such example where you're basically changing a C into a T, and then mismatch repair will half of the time repair the opposite strand into an A, and sometimes we'll repair this strand into a C, and so on and so forth, okay? Everybody with me here on sort of true base editors rather than cutters, nucleases, yeah? So basically, again, there's a huge amount of research into finding additional base editors. Instead of having only a C to T mutation, you can induce, you know, many different types of mutations. So there's a lot of work going on right now by, you know, David Liu, for example, at Harvard, and also here at the Broad, for, you know, finding new enzymes that can be coupled with a CRISPR system to then directly induce genome editing rather than cuts. And then, of course, the applications are endless. And, you know, here's, for example, one example where a Cas9 mouse has been generated that actually already has Cas9 in the whole CRISPR machinery. So all you need to do is just give it different guide RNAs, and boom, you can edit whatever you would like. You know, this is an incredibly efficient system. Everybody with me on this? So that allows you to now rapidly generate genetically engineered mice by basically selecting the cells that we're editing and then creating the mice from those cells that we're editing. You can use that to model autism, for example. You can sort of carry out mutations that recapitulate autism in, you know, specific alterations. You can, you know, target, for example, PCSK9 to reduce cholesterol. You can basically, you know, target that in a mouse and then show that indeed cholesterol is dramatically reduced. You can use screens with Cas9. You can basically, you know, use Cas9 knockout screens to identify genes involved in resistance. For example, you could use different guide RNAs and then select those cells that showed resistance, and then sequence to figure out what actually changed in their genome, and then identify the genes whose alteration or whose knockout was in fact leading to that change. So you can basically, you know, engineer the system to, instead of targeting one very specific site, create a whole library of guide RNAs, and then use that to target effectively the entire genome by chance, and then figure out, working backwards, what did I actually cut, and then how does that allow me to understand the, you know, the underlying machinery. Cas9 is an endonuclease. Yeah. I mean, it's been used for many different applications. You know, let's talk after class. Yeah, so let's talk about sort of the specific applications right after. So this is all based on the cutting of Cas9, the nuclease. But dCas9 or dead Cas9 is a non-cutting Cas9. So it does all of the above of sort of recognizing the PAM site, using the guide RNA, locking onto a very specific site in the genome. And then it just sits there. And then you can couple dCas9 or dead Cas9 with all kinds of other stuff. You can basically use exactly all the stuff that we saw for the zinc fingers to bring in activation domains or inhibition domains, and do CRISPR-I or CRISPR-A, which turns on genes or turns off genes. And you can do pretty much anything. So basically, Cas9 can be used as a nuclease for genome editing. It can be used as a screening approach. It can be used for transcriptional control using dCas9 with activating or repressing domain. And we focused a lot on sort of gene surgery. That's sort of what the popular press has focused a lot on sort of being able to cure diseases by going into either the germline or somatic tissues and changing genes. But it can also be used to, target bacteria, or it can be used to target viruses, or it can be used to create new drugs, or it can be used to create new animal models that are much more effective, such as the mouse that actually has the CRISPR system and Cas9 already there, or to understand genetic variation, or to create new biomaterials, or to actually, and that's a huge, huge industry, to create more insect-resistant food, or parasite-resistant crops, and so on and so forth, or even to create biofuels. So this is one tiny part of a huge, huge industry of applications across medicine, basic biology, and biotech for using these kinds of systems much more broadly. So what did we talk about today? We talked about ways to rewrite genomes. So we talked about ways to carry out synthesis of DNA segments in very, very high throughput and its application to understand and rewire gene regulation, modification of this technology to basically increase its application by many orders of magnitude each time. And then we talked about this whole general principle of endogenous genome editing. First, endonucleases, such as zinc fingers and TALENs, for basically being able to target different regions of the DNA. We talked about the repair pathways, both non-homologous end joining and this homologous repair, and the diversity of outcomes that can happen in particular regions. And then we talked about this incredibly revolutionary technique of CRISPR-Cas9, where instead of having to create these overlapping protein domains of zinc fingers or these long amino acid arrays for targeting individual nucleotides, of basically using an RNA guide for this. We talked about the origins of the CRISPR-Cas9 system in bacteria as a defense mechanism, the discovery of the system, the optimization of the system, a variation of this that actually carries out base editing rather than genome vandalism, and various applications. And we talked about therapeutic applications, ways of multiplexing this and sort of screening with thousands of guide RNAs across the genome. We didn't talk too much about delivery, but basically being able to now take this and deliver it into individual cells is, a big, big deal. And also a lot of sort of next generation tools are on the way. You may have heard the news about this scientist in China who actually claims to have editing these girls that were recently born with their genome edited. I mean, there's a huge number of ethical implications surrounding this. How can you consent an unborn baby, or how can, were the parents truly informed, were there, were the IRBs in place for actually doing that, and so on and so forth. So like any technology, it's extremely powerful, but it's also extremely dangerous. And I think it is a huge, huge area of investigation. The possibilities are endless. The technologies have already matured many, many generations over, and you can just use that to now, follow your imagination, basically, truly engineer new biological systems and asking new biological questions and so on and so forth. Who feels that they've learned something today? Yay, awesome. So that was the last of my lectures. Next week, it's you guys, and I look forward to that. Thank you. Thank you.